So in our group, we have recently finalized a study where we were searching for lytic phages inside bacterial assemblies. And you'll hear about the study tomorrow from Thomas, my supervisor. But we had to analyze a very big data set of 3.5 million bacterial genomes. And for that, we needed a tool that would work very well. And it was important for us that the tool would be also fast because we have deadlines. And so we used our own program called Phager, and it's quite simple. It's just made to recognize phage from bacteria. And it's a machine learning based tool. We used over 11,000 phage genomes and over 50,000 bacterial contigs to train our model. And to process, here's a quick overview of how we processed our data. So we had our phage and bacterial genomes as an input, and we did gene calling on them. We removed prophage regions from the bacterial genomes. We generated features for each gene of each genome, and those features basically describe each gene in different sorts of ways, like a GC contact and molecular weight and length and all sorts of things. I'll mention them later. And then we fragmented our genomes into such 3 gene fragments where we have information collected about the middle gene and information about neighbor genes. And then we labeled our triplets as which ones are from phage and from bacteria. And then our model had to learn what is phage from that. And what I really like about machine learning that you can actually extract how important and which features were the most helpful to recognize phage for our program. And here I highlight only first top most important features that our phager decided that those were the most helpful to recognize a phage. So the number 1 was protein length, and this one is a bit boring because we expect that proteins of phage are usually, protein sequences are usually shorter on average. But I thought interesting ones were percentages of amino acids leucine, tryptophan, and isoleucine. It's because we had, in our list of features, we had percentages of all the amino acids, but those were the most important for phager. They were most helpful for phager to say that, okay, this is a phage. I don't know if the proteins tend to have less or more of those amino acids. We're going to look into it in the future. But then there was also another feature that was important, hydrophobicity of a protein. And I heard that phage proteins are more hydrophobic on average, but I'm not sure if I did enough research on that, but you could probably conclude yourself. But this is very interesting to me. So in a nutshell, how the phager works, it gives out a probability of this small 3-gene fragment to be a phage. So here we have a bacterial genome on the top and phage genome on the bottom. And the bars are the triplets. And if it's red, it didn't pass this phage threshold. And so you can see that mycoplasma genome is mostly red. And that's, so phager says it's not a phage. That's what it does. And I tested our model on so-called validation set, where we had a collection of bacterial and phage genomes that were not present in the training. And those genomes were also not closely related to the genomes in the training set. And here you can see the percentages of correct predictions for the bacteria and for the phage. And you can see that phager predicted over 93% of phage sequences as phage, which is really good, and over 99% of bacteria sequences as bacteria. So we're happy with this result. And I compared our phager with other popular predictors. And here on this graph, you can see MCC. It's a Matthew score. It's a prediction score, which accounts to false and correct predictions. And the closer to 1, the better. And you can see that phager is comparable to 2 very good prediction tools, Vibrant and VirSorter. But phager has a great advantage. It's the speed. So this how much genomes per hour phager can produce is over 5,000 genomes. that is 10 to 11 times faster than the next fastest in the selection tool, GenomeAd. So using Phager saved us, I think, over 3 months, if not more, time when we used Phager compared to other tools. So you can give a try to Phager through this link on GitHub. I'm currently working on a beta version that's going to be uploaded soon. And I thank my supervisor, Thomas, for help and support. And to Marta, they're here today. Thank you.